So today in class, guys, we were talking about sort of the history of the discovery of DNA and kind of how it got to be known as the genetic material. So what I wanted to do is give you guys a short video here about a few basic experiments that were run and a few basic discoveries so that you guys kind of understand where it comes from, and hopefully it just gives you a better understanding of DNA in general. So here is the story of DNA. First off, we want to talk about genetics and it's DNA's connection to genetics. They've known for many, many years that, there is this heredity, that traits are passed on from Mendel's basic experiments on pea pods to, animal husbandry, where they're taking different animals and mating them together to get an offspring that has certain traits. Genetics was known. They just didn't know what caused that hereditary, that passing on of information. So kind of the question of how does genetic works, I'm being a little bit funny here, but for a long time, up until 1869, the answer was I don't know. They didn't have a definite, what was causing this to happen. And then around 1869, you can see here that Miescher, he discovered DNA. And I mentioned this in class, but he was studying white blood cells in pus of infections, and he was able to isolate this material, which turned out to be DNA. He didn't know, exactly what it did, but he was able to isolate the DNA and the proteins that were in the nucleus of the cells. Years pass, and then in 1928, and this is probably one of the first kind of major, right here, 1928, one of the first major experiments was done by Frederick Griffith, and it kind of gave us this idea of, well, maybe we do have a little more knowledge or understanding of what it was. What Griffith did, here's an overview, and this is from your textbook, guys. If you look in your text underneath the DNA section, you'll find some information in this image. But basically, Griffith was studying two different forms of pneumonia. And what he did is he took the S form, which was the deadly form, and then the R form, so smooth and rough is what they were. The smooth form was deadly, the rough form was not. And he basically ran a pretty simple experiment. Thing one, which was right here, he took the S bacteria, the deadly bacteria, injected into mice, and lo and behold, the mice died. That makes sense. He then took the R form, which was the not deadly, injected into mice, and lo and behold, the mice survived. Again, expected. Then he took the S bacteria, the deadly form, he killed it, he heated it, which killed the bacteria, injected that into live mice, and then they survived, which makes sense. If you kill the bacteria before you inject it, it shouldn't kill the mouse. And then this is where things got a little bit weird. He took the heat-killed bacteria, which did not kill. He took the live R bacteria, which did not kill. He mixed them, and then he injected that, and it turned out to kill the mouse. So what he kind of figured out is that there was he called it a transforming factor. There was some material that passed between the dead bad bacteria and the live good bacteria, and it transformed or caused them to become a deadly bacteria. Well, what does that say? It basically said this, right? Something in there. There was a transformation. There was some material, something passed between them, some sort of genetic information, if you will, to cause them to change from harmful to harmless. That was his experiment. That's basically what it said. It didn't get into what it was. It just said that there was a transforming factor. All right, so that was Griffith. Years later, in 1944, three scientists, Avery, McCarty, and McLeod, repeated those experiments, but they added on to them. Much of science is taking previous experiments and results, retesting, changing your variables, changing your controls, and seeing if you can get a little bit different to work. So what these guys did is they took the same bacteria, but they removed the lipids and the carbs, which basically proved that lipids, cross that out, and carbs, cross that out, were definitely not the genetic material. Then what they did in consecutive experiments, they would add a destructive enzyme to Griffith's experiment, And the reason they would do that is if they destroyed, let's say, the proteins, and then they ran the experiment and the transformation still happened, then it meant that the protein was not what was causing the transformation because it wasn't there and the transformation still occurred. So they went through and did that. They already knew it wasn't lipids and it wasn't carbs. So what they did is they added a proteinase. Remember, ASE at the end of the word there, that means it's an enzyme. Proteinase destroys proteins. And they ran the experiment, and that transformation of good to bad bacteria still occurred. Therefore, proteins were not the transforming material. They also added ribonucleases, which are an enzyme, or they are enzymes that will destroy the RNA. Transformation still occurred, so therefore they knew it was not the RNA. Then they added a deoxyribonuclease, which again is another enzyme that can destroy the DNA, and there was no transformation. So what that said to them is it was fairly good proof that by removing the DNA, the transformation of good to bad bacteria did not occur. Therefore, the DNA was in fact the thing that was causing the transformation. So DNA was the transformation factor. Again, the proof is pretty good, but the problem was that the scientific community was still skeptical. They looked at it, they retested, it made sense, but they needed a more definitive proof. So again, you can see now we've gone another handful of years here. We're going to take a look in the 50s, at 1952. Two scientists, Martha Chase and Alfred Hershey, they now took a slightly different tact. They use these things called bacteriophages. They're a virus. Remember, the phage word means to break or to eat. So these are viruses that eat bacteria. They destroy bacteria. They look like little lunar landers, I think, but basically viruses are super simple things. They exist with two parts. They have a protein outer coat and a DNA inner core, and that's all they are. So you've already broken it down to proteins or the DNA. Now, common knowledge at the time, proteins are so complex and so diverse that the people thought, most of the scientists thought that DNA was not the genetic material and that protein had to be, just because it was so much more complex. So what they did is they wanted that answer. Is it the protein or is it the DNA that causes these transformations? Understanding how these bacteriophages work, they knew that they could determine that with a fairly simple experiment. It's often called the blender experiment. But basically what they did, I'm going to keep it fairly simple. They injected two different radioactive markers. One of them was radioactive phosphorus-32, and one of them was radioactive sulfur-35. It's a neat setup because DNA contains phosphorus but not sulfur, and protein contains sulfur but not phosphorus. So by tracking these radioactive markers, they could tell if it was DNA that entered the bacteria or if it was the protein. So you can see the results in the image here. When they injected the phosphorus, which came with the DNA, they went through, and what they discovered was in that bacteria at the end, it was radioactive, which meant that the phosphorus entered the cell and transformed that bacteria. Remember, phosphorus is only found in DNA. This one below, they took sulfur and they had it marked. It went through, and the sulfur did not. So there's no radioactivity, so that radioactive sulfur did not enter the cell. Therefore, they knew that it was not the sulfur. Therefore, it was not the protein. Gave absolute definitive proof that DNA is the transforming material or the genetic material that is passed on. It is not the proteins. So now, 1952, they really have the knowledge that DNA is in fact 100% the genetic material. So at the same time as all happening, there was also work on the structure, which we've talked about a bunch in class. The basic names I wanted you to really know here. These were two names, Maurice Wilkins and Rosalind Franklin. We saw the little video clip about where Rosalind Franklin was sort of the unknown in this. But basically, these two were both involved in X-ray crystallography, where they take stretch out DNA molecules, crystallize them, and then they shoot X-rays through them and they get images. Now this image right here doesn't mean a lot to us, but to people that know X-ray crystallography, that shape, that sort of X shape like that, is not the actual, they're not taking a picture of it, but they're taking a picture of X-rays that have passed through it and then diffracted onto the plate. And that, to anybody who knows this material, that screams that it's a helical or helix shape. And that's the best picture they ever used. That's the one that got used by Watson and Crick to really help them determine that that's the kind of structure of DNA. So Franklin and Maurice Wilkins, if you remember, he received a share of the Nobel Prize with Watson and Crick. Rosalind Franklin passed away from cancer, which was more than likely caused by all the X-rays she worked with for her career. So she's not as well known, even though she was kind of the driving force behind the proof of what the DNA structure was. Two gentlemen, I'm sorry, before I get to that, the image of the X like that, if you look here, is a basic idea of what they do. They shoot an X-ray through a crystallized structure of DNA, and it shows on the screen or on the plate. That twisted look is representative of a helix, and the equal spacing of the sort of the gaps shows her that there was equal spacing of those rungs of the ladder. And that, in and of itself, used by Watson and Crick. They saw her photos. 1952 into 1953, they constructed a model, and that model is what we currently know is the DNA double helix with the phosphate sugar backbones and the rungs made of A to T or G to C nitrogen bases. So I just wanted to, again, give you a quick thing. If you want to go back and review those couple labs, make sure you understand how the removal of the different, the proteinases, the ribonuclease and the deoxyribonuclease kind of proved that it was DNA, and then how further proof was gotten by Chase and Hershey with their work with the bacteriophages and the radioactive markers. Thanks, guys, for listening, and we will see you on Monday. Stay warm.